We start this laboratory session, IVF laboratory session, with these small groups. It's quite a small group, actually. I expect it to be more. However, I would like first to welcome my dearest friend and colleague, Professor Izzat Sobki, is the father of genetics in Egypt, and Dr. Muhammad Hadi. He's an old friend, and he's one of the very good and famous embryologists in Egypt. This session we have 1, 2, 3, 4, 5, 6, 7, 8, 9 speakers. Professor Izzat Sobki will start, and of course you are all eager to listen to his experience and science, and you know all that Dr. Izzat Sobki, he pioneered the genetics in Egypt since around 100 years. His lecture is the role of genetics in reproductive medicine. تفضل دكتور عز.
Bismillah ar-Rahman ar-Rahim. Thank you very much, Professor Abdul Hamid Wafi, and thanks very much, my dear friend Muhammad. For this next 15 minutes, I will try to give you a very simplified summary of the progress in the field of reproductive genetics. I will divide my presentation into a very single slide, giving you the timeline. Then I will go through the technical aspects in the field of reproductive genetics in general. Then the application or implementation of different techniques. As you all know, talking about medical genetics means that we take the clinical data and we move inside not only the cell, but inside the nucleus to verify the underlying genetic etiology. So we should consider that the human genome or the genetic contents of a human cell is simply formed of the chromosomes. And actually, these chromosomes are made up of chromatin fibers. If you stretch these chromatin fibers, you end up with the double helix DNA, which is formed of nitrogenous bases, and this form actually the genes. So either you study the genetics at the level of the chromosomes, and this is what we call it cytogenetics, or you go and sequence or read the nitrogenous bases of the DNA. That means sequencing of the DNA, and you are talking about molecular genetics. So based on this very basic knowledge, we can say that any genetic disorders could be classified into 3 groups, either due to chromosomal abnormalities or a single gene disorder. And chromosomal abnormalities you are all aware of: the aneuploidies, the trisomies, the monosomies, the translocations. For single gene anomalies, those are the groups that every now and then you hear about such an autosomal recessive disorder like beta thalassemia, the most common genetic disorder in this area, or Duchenne muscular dystrophy as an X-linked, or hemophilia as an X-linked, or dyschondroplasia, those very short skeletal dysplasia, and this is autosomal dominant. So you cannot offer chromosomal study for single gene anomalies. You have to ask for DNA study or DNA sequencing to verify the underlying abnormality, and in that time we call it mutation. So you have to pinpoint the underlying mutation in the gene. And another group is what we call it polygenic or multifactorial, that you have an underlying genetic susceptibility that react with certain environmental factors to precipitate a genetic disease. The most common multifactorial disorder is neural tube defects, meningocele, meningomyelocele, anencephaly, congenital heart diseases. Most of the congenital heart diseases are due to multifactorial disorders. So you cannot say that meningocele, meningomyelocele as an isolated abnormality is dominant or recessive or X-linked. It's a multifactorial. And most important environmental factor in this group is the folic acid. That's why we give folic acid very early. And I should state in here that you should know that the neural tube closed by the day 26 during embryogenesis. So before the lady telling you that she is pregnant, actually the neural tube closed. That's why we should give folic acid preconceptionally. If you describe folic acid, so that needs some sort of national awareness. That's why they fortified the cereals abroad in Europe and USA with folic acid. If you look to the time table, we should realize that between 70s and 80s, the recent cytogenetics developed, doing chromosomes with high resolution and different application, amniocentesis and bone marrow chromosomes, all these conventional cytogenetics. Then by the 90s, early 90s, the FISH technique or fluorescent in situ hybridization, the combination between molecular genetics and cytogenetics developed at that time. And then by the 2000s, the CGH and its application started. And almost 5 years ago, the first delivery of a baby after BGS or pre-implantation genetic screening using the microarray technique. Conventional cytogenetics, that you take a sample, you start a culture, and you harvest this culture, and you can have a karyotyping. You see the 46 chromosomes, and you can detect any abnormality like that. And you can say that this is XY or XX, or having a deletion or inversion or this is the conventional cytogenetics. But having a molecular technique to detect a specific tiny area on the chromosome using a DNA probe, this is the in situ hybridization, the FISH technique. Since we are using a reporter fluorescent molecule, we call it fluorescent in situ hybridization or FISH technique. And the advantage of using the FISH technique, that in conventional cytogenetics you have to have chromosomes. But actually with the FISH, you can study the chromosomes and still you can study the interphases. If you don't have metaphases, still you can have an idea about the interphases. And the immediate application of that was direct FISH application to uncultured amniocytes. Before that, we have to take amniotic fluid and induce a culture for 2 weeks. Then we have karyotyping. But with fish, you can take directly few cells of the amniocytes, put it on the slide, you apply your fish probe for chromosome 21, 13, 18, X and Y, and in 24 to 48 hours, you can have an idea about if this fetus is male or female, having Down syndrome or not. So that was at that time a revolution. Moving with the fish technique and applying it to a single cell. If you have a single cell, and you can still have an idea about the chromosomal content of this single cell as a representative of specific tissue. And then the number of fish probes that you can use it on a single cell increased to 11 fish probes. So if this is a Palestinian family represented to us with multiple fetal losses and fetal malformation. We tried to find out the cause, but using the microarray, we detected a very tiny translocation that you will never detect it by a conventional technique. Then we offer the family a prenatal or PGD based on verifying the etiology. So taking a good history and putting this clinical data with the history using the family pedigree, this is the family tree, the family pedigree. So we construct a family pedigree to verify the mode of inheritance of such abnormality. If you move to a very, very detailed study of the sequence of the DNA, this is the molecular genetics. And since the human genome, which is formed of 23 pairs of chromosomes, is actually written by 3 billion letters of nitrogenous bases, so you have a lot of genes, almost 35,000 genes. And to identify these genes, actually, when you look as if it is the book of our life, you have an initiation codon and termination codon. As if you have a chapter, you have a starting page of the chapter and the end of this chapter. So we verify the initiation point and termination point, and we put a markers as if you are in the library and you need to have copies of a certain paragraph. So you identify from page 101 to page 110, you give it to someone to make a photocopy of this area only. So the basic technique of a single gene study is to have the gene, you know the initiation codon, the termination codon, you put a marker, then you amplify. This amplification is the polymerase chain reaction, the PCR. Okay, so then when you have multiple copies of this gene, you can study the gene and to see if there is any mutation or not. So we can summarize that you can look morphologically to the cell, or you can study the chromosomes, or you use the FISH technique, or you can sequence the whole genome. Nowadays, there is a test called whole exome sequencing. You take only 2 ml of blood on editor, and you can study 22,000 genes in a single test called whole exome sequencing. You read the whole genome of yourself from a tiny sample of blood. Actually, the genetic disorders, as we mentioned, chromosomal, single gene, or multifactorial, and if you look to the incidence of congenital anomalies, it's almost 3%. And if you divide this 3% equally, then you would think about you have equal portions of chromosomally abnormal offsprings and with others with single gene anomalies. But when you take it to the practical, daily practical lab, you find that 90% of the load of the lab is cytogenetics and 10% only molecular study, and this needs to raise the awareness of single gene anomalies and the possibility of diagnosis of such abnormalities. This is different translocations, and you can have the probe specific for this translocation, and you can offer prenatal or PGD using a specific probes for such a translocation. Actually, the case will be presented to you with infertility or recurrent fetal losses, or fetal losses with fetal malformation. You offer conventional cytogenetics, you detect the abnormality, then you discuss with them the reproductive options, either to go for prenatal or pre-implantation diagnosis. The pre-implantation diagnosis using FISH technique needs a technical experience to have good spreads of the blastomeres, because if you have the blastomere very nicely spread like that, it's very easy to the molecular probe of FISH technique to penetrate and to hybridize to the target DNA and give you an excellent signal. If the spread is not good, then you have a problem. The other part is the molecular or single gene anomalies. This is actually a case that a man has a hemophilia and he has 2 daughters. So since he is hemophilic, then his Y is normal, and he carries the factor 8 deficiency on the X. So all his boys will be normal, while the daughters will be obligatory carriers. We offer the 2 daughters PGD, and because they don't want only a female, they need whatever female is which is not a carrier, or a male who is not affected. So we offer them PGD. And that time you don't study only, since they are not interested in sex selection, you go for the gene itself and you offer them DNA sequencing. So there is a progress in the technical aspect to increase the application of different genetic techniques in the field of assisted reproduction. The FISH technique with its revolution still has limitations, because you can only offer 11 FISH different colors probes, and this creates a problem also because it depends on the examiner. If you look under the microscope, you have different colors, you can have mistakes, or you can have overlapping of different colors. So this is one of the limitations of FISH technique. Then looking forward to improve our technology and sort out this FISH problem that you have limitations, we looked for the microarray, and the microarray moved to the what we call it single nucleotide polymorphism to have a very high resolution level. And applying the SNP genomic coverage, you can study, as you can see, not only one probe for each chromosomes, you have thousands of probes on each chromosome. So you have a very high detection level of different chromosomes. And this created that we need to improve the implantation rates in IVF and ICSI. So we depends, or embryologists depend on the morphology of the blastomeres or the embryos by day 3 or day 5. But actually, if you look to this morphology, and I ask you which one is the best one, and then you offer a microarray or 24 chromosome study, you will find your selection is different based on morphology from the selection based on the chromosomal contents of each embryo. And this is the reporting of the chromosomes. It's not anymore karyotyping, but it depends on bioinformatics. and a curve to give you if you have 1 copy, 2 copies, or single copy of each chromosome, or even partial deletion of 1 chromosome. But still, again, we have a limitation of each technique. If you use the microarray, you cannot detect balanced translocation. You cannot detect point mutation for single gene. You cannot detect the triplet repeat expansion like in cases of Fragile X syndrome, and limited ability to detect polyploidy and limited ability to detect mosaicism. So we looked for a technique that can cover this molecular study with the chromosomes. And actually the solution was the next generation sequencing. Using a next generation sequencing, you can study the 24 chromosomes, and you can study single gene anomalies, and you can customize even multiple genes to study. So you can screen or detect different genes. At the same time, you can have a chromosomal profile for the 24 chromosomes. And this is a very highly sensitive technique, very fast. You can study all this in 12 hours, and you can use a lot of samples. You can run 24 samples in a single run. It's a very simple workflow, and the reporter is very good to detect very simply any abnormalities at the level of the chromosomes. And this is a different indication for BGD and BGS, recurrent miscarriages, unsuccessful or ICSI failure, repeated ICSI failure, advanced maternal age, severe male factor infertility, or if one of the couple is a translocation carrier, and if you want to study also a single gene disorder at the same time. So now we have multiple or different publication for a success of normal pregnancy after NGS or next generation sequencing. Still, the CGH or microarray is struggling and fighting with the next generation sequencing. FISH is still surviving, and we're looking forward for a new technology to offer for our patients. I would like to thank my team at the Medical Genetics Center, and thank you very much.
Thank you, Professor Izzat, for this very nice lecture. And actually,
it's my pleasure to co-chair this session with elegant, he said I'm the father of genetics. He is the pharaoh of the Egyptian embryologist. If you go to the pyramid area, you will find the fourth pyramid is Abdul Hamid Wafi. And it's our pleasure all together to listen to his presentation, embryo assessment, old versus new methods.